Hello. Today we'll start our lecture by differentiating between how the secreted molecules inside the organism can mediate three forms of signaling. So we have a paracrine signaling, and we have a synaptic signaling, and we have an endocrine signaling. These are three majorly different ways of signaling or communicating between different parts of the organisms. So in the paracrine signaling, the cell secrete the secreting molecule, and this will have a local effect on the neighboring cells. So it is a local effect. That's why the secreted molecule is called local mediator. While in synaptic signaling, it involves a neuron or nerve cell that transmit the impulse on relatively long distance until reaching a target cell, in which this nerve impulse will be, this electrical impulse will be transformed into a chemical signal and pass through the synapse between the end of the neuron and the target cell. And in these cases, the target cell could be a gland or a muscle. In the third type of signaling, which actually what we are dealing in that course, is the endocrine signaling, in which the endocrine cell will secrete a hormone, and this hormone will pass into the blood, carried by the bloodstream over a relatively long distance until reaching as well a target cell. So you need to know that the endocrine signaling is the slowest type of signaling among these three types of signaling. And as well, the nerve cell is the fastest to transmit the information by electrical impulse that can reach around 100 meters per second. Once it reaches the end of the neuron, this nerve electrical impulse, as we said, is changed to chemical molecule represented by a neurotransmitter from this nerve terminal end that diffuse through a space that have like around 100 nanometer in size to the target cell. While the hormones are greatly, since they are moved through the blood, so they are greatly diluted in the bloodstreams, while the neurotransmitters are less diluted because they are locally concentrated at the region of the synapse, and they can achieve a high local concentration at the target cells for the neurotransmitter, which is a major difference between the neurotransmitters and the hormones. Of course, any of these secretory molecules, either the paracrine or the synaptic or the neurotransmitters or the hormone, need a specific receptors, but there is a difference between the receptor of the neurotransmitter from the other receptors, because the receptors of the neurotransmitters on the target cells have to be relatively of low affinity of the ligand. That's what's meaning of low affinity? That's meaning that your neurotransmitter can attach and dissociate rapidly from the receptor in order to terminate the response. You need to think that if the receptor of the neurotransmitter have a high affinity for the neurotransmitter, so the effect of the nerve will be long-lasting, and this will not be good for the organism. So again, if you look closely to the signaling, synaptic signaling, you can see three different neurons. Each of them, the end of the neuron can sit on a different type of cells depending on the neuron. And you can see each type of neuron, A, B, C, coordinate and send the signal to a specific type of cell. So you can see neuron A can affect the target cell A, and the neurotransmitter is formed at the junction between the end of the neuron and the target cells. As well, you can see as well nerve B have a specific type of cells, and nerve C have a specific type of cells. So you have various target cells depending on the various neurons. As well, the same case for the endocrine signaling. You have three different endocrine cells that secrete three different hormones, and each hormone has a specific receptor on each specific target cell. So you can see the major difference from this diagram, that the signal which is conveyed by the endocrine signal is much diluted. So you need a high concentration of hormone is secreted to the blood in order to make sure that this hormone will find its target cell. So we'll talk now about the autocrine signaling, which can coordinate the decision by a group of identical cells. So what's the meaning of autocrine signaling? It means that cells can secrete a secretory molecule or a signal that can affect itself or can affect cells nearby. So it's a kind of a paracrine as well, but once it affects itself, it's said autocrine. So what this type of signaling is very important during development, because this autocrine, and auto means self. So this autocrine is used to encourage a group of cells to differentiate to the same developmental decision. Say, for instance, if cells of the embryo need to be differentiated into, for example, kidney cells, there will be one cell that secretes a signal that will affect nearby cells or itself to give a general developmental decision for this group of cells to differentiate into a kidney cell, for example. So see, you can see in this diagram how this cell can secrete this secretory molecule represented by the red dots. And this secretory molecule will have a receptor on the same cell. And you can see initially the cell with green nucleus is different than the cells with brown nucleus. Once this signal is released from this cell, it will affect the nearby cells. So all these nearby cells will behave the same as the cell which secreted initially the signal, the red signal. And you can see as well that each of these cells will express a receptor that is specific for that signal, making all these group of cells to have the same developmental decision, or for example, to be differentiated into the same type of cells. شباب في حد عنده سؤال في الجزء اللي فات؟ شباب سامعيني؟ في حد عنده سؤال في الجزء اللي فات؟
كله تمام دكتور.
Okay. So let's here see, generally there are four methods of cell-to-cell communication are found in human body. It ranges from a direct communication into remote communication. If we're looking into direct communication, you can see that ions, small molecules, can transfer between two nearby cells. And this will lead to a direct movement. Either the movement can be occurred through the gap junctions. And this gap junction actually is connecting the cytoplasm of two adjacent cells. And the example, the famous example of this type of direct signaling or direct communication is the ion flowing between the cardiac muscle cells. And you need to understand this is a very important way of communication, because the heart muscle or the cardiac muscle need to be synchronized in its action of contraction and relaxation. So you can see there is a direct communication between each muscle cells in our heart. The second type is the synaptic, as we have discussed, and the chemical messenger in that case is the neurotransmitter. And as we said, the mechanism, the neurotransmitter move through the synaptic cleft in order to affect the target cells. And one of the famous neurotransmitter is acetylcholine. The third type, as we said, the paracrine, and you can see that cells, the secreting cells will secrete this green signal, which act as a local hormones and affect the nearby cells. One of the famous examples of the paracrine signaling is a molecule called prostaglandins. It's actually a group of molecules called prostaglandins. And this signal will be secreted through the extracellular fluid to the nearby cells. And of course, each of these signals will have a receptor on the target cells. For the endocrine communication, we have discussed that the endocrine cell will secrete the hormone into the bloodstream. Of course, one of the major hormones is the insulin, and it will be carried by the blood either to a nearby or a distant, relatively distant cells, which should have the receptor for that hormone. So let's classify types of hormone. So basically, the hormones are classified into three different, based on three different criteria. According to the anatomical source, so according to where this hormone comes from, according to its chemical composition, and according to its solubility, the hormone, how it's soluble. Is it lipid soluble or water soluble? According to anatomical source, you will see that these hormones are secreted from a major gland in our body, which is called the hypothalamus, like the growth hormone releasing hormone, the growth hormone inhibiting hormone or somatostatin, the gonadotropin releasing hormone, the corticotropin releasing hormone, the thyrotropin releasing hormone, prolactin releasing factor, prolactin inhibiting hormone or prolactin. And of course, we will deal in details later on for these hormones, and these are hormones secreted from the hypothalamus. Other hormones are secreted from the pituitary gland, and you need to know that the pituitary gland generally is divided into two major regions, the anterior pituitary gland and the posterior pituitary gland. The major hormones of the anterior pituitary gland are adrenal corticotropic hormone, corticotropin, the thyroid stimulating hormone TSH or thyrotropin, the melanocyte stimulating hormone, the growth hormone, very famous growth hormone or somatotropin, the prolactin, the gonadotropins, which include the FSH and LH, the follicle stimulating hormone and luteinizing hormone, while the posterior pituitary gland only secretes two types of hormone, the ADH or vasopressin or antidiuretic hormone, same name, and the oxytocin. And these actually two of the important hormones and only secreted by the posterior pituitary gland. Other hormones are secreted from thyroid gland, parathyroid gland, adrenal cortex like the steroid hormone, adrenal medulla, hormone like adrenaline or noradrenaline, or epinephrine or norepinephrine. Hormones are secreted from the kidney, like the renin, angiotensin 1, erythropoietin. Hormones are secreted from the pancreas, like insulin and glucagon, and of course from the gonads, like the sex hormones. What about the classification of the hormone according to chemical composition? So actually, we have either the hormone are peptide hormones, so that's mean it's a formed from it is mainly protein in nature. So these polypeptide hormones include all the anterior pituitary gland hormones, and as well all the anterior pituitary-like hormones that are generated from placenta during embryogenesis, and as well pancreatic hormones. And as well the parathyroid hormone and calcitonin, the kidney hormones as well, while there are some other hormones which we call it oligopeptides. That means it's short proteins which consist of few amino acids. That's meaning oligo. Oligo means few. So hormones, the two hormones of the posterior pituitary gland, which is the ADH and the oxytocin, are secreted from the posterior pituitary glands. And as well the hypothalamus hormone, except the prolactin inhibiting hormone, which is the PIH. Let's talk closely, let's go closely into how these polypeptide hormones are formed within our cells, especially the endocrine cells. So initially you have the nucleus, and you have inside the nucleus, you have the DNA, and you can see the nucleosomes. and the nucleosomes, which actually DNA wrapped around histones. Initially, RNA polymerase, the enzyme responsible for transcription, bound to the DNA. It transcribed the messenger RNA precursor, the initial molecule of the messenger RNA, which soon after that it undergo a process of maturation or processing, RNA processing, until forming the functional messenger RNA. Once the functional messenger RNA is formed, these migrate outside to the cytoplasm. You need to know that before the migration of the messenger RNA from the nucleus to the cytoplasm, it needs to be modified. So this messenger RNA will acquire a cap, which actually consists of methyl 7-guanylate triphosphate, and it will acquire as well a polyadenine tail. And basically, these two modifications is to protect the messenger RNA from degradation in the cytoplasm. And as well, the non-coding region of the messenger RNA will be removed. This non-coding region is called introns, will be removed before going out to the cytoplasm. So generally, there are three modifications occur to the messenger RNA precursor in order to form a functional messenger RNA. These three modifications include a cap, addition of the cap at the 5' end, and addition of a polyadenine tail at the 3' end, together with the accession of introns, which is the removal of the non-coding regions. Once inside the cytoplasm, this messenger RNA got recognized by the ribosomes that is attached on the Golgi region, and it starts doing together with the help of the transfer RNA as well, which recruit the amino acid, it will start translating the protein. And you can see that the protein initially is formed in the cisterna of the endoplasmic reticulum. You can see that the hormone, which actually the polypeptide, is initially formed, and it is called pre-pro hormone. It is still non-functional hormone, and it needs to be processed in order to be a functional hormone. You can see that inside this endoplasmic reticulum, it is converted from pre-pro hormone into pro hormone. Basically, a region of that hormone is excised or removed in order to be a step further in the process of maturation of that hormone. Soon after that, by the addition of a carbohydrate moiety or CHO carbohydrate, you can see that the prohormone is converted into mature hormone. This mature hormone, once is matured, is packaged inside a vesicle after this process of post-translational. And what's the meaning of post-translational? That means it is the addition of a molecule like the CHO on amino acid on the translated protein. That's why it's called post-translation, because the modification occurred after the translation of the protein. Once it is mature and got all the post-translational signal needed for its function, it will be packaged into secretory vesicle, and it will be secreted and transported in the blood until reaching the target organ to have its biological function. So you can understand here that DNA, there is a transcription to form a pre-messenger RNA. There is a post-transcription process that what we have said is the conversion of the precursor messenger RNA into functional messenger RNA. Once the messenger RNA is inside the cytoplasm, it will be translated into pre-protein. With further modification, it will be converted into functional protein that is transported via the blood to do its biological action. And this actually the mechanism of synthesis of polypeptide hormones. في حد عنده سؤال يا شباب؟
ماشي الحال دكتور.
Okay. So let's see more closely what's happening in the process of transporting and secretion of these polypeptide hormone from the protein secreting cells. As we said, the protein is initially formed in the rough endoplasmic reticulum. You can see the ribosomes here represented by the ribosomes represented here by the purple on the surface of the cisterna. And you can see that the proteins are formed inside the cisterna. So in this rough endoplasmic reticulum, once the proteins now are loose inside this rough endoplasmic reticulum, secretory vesicle started to evaginate, and this secretory vesicle will evaginate and start moving toward the smooth endoplasmic reticulum and Golgi apparatus. And you need to understand in this transition state or transition elements, and the function of the Golgi complex actually is to sort and package, and forming the secretory vesicle. You can see now secretory vesicle are formed with the functional protein, which actually the hormone, and this functional protein, initially it will be immature secretory granules, and then it forms mature secretory granules after some processing, and soon after that it can be released outside to the blood stream. You can see that some secretory vesicles as well can go away without secreting anything, and you can see as well some of the secretion if the protein formed after the process of translation is malfunctioning, and actually it is not a proper hormone. It is directed toward the lysosome, and of course, you know, the lysosomes contain a lot of enzyme. It will degrade this protein, and it will prevent that protein from going out to the circulation. Because if it's released to the circulation, it will be a malfunctioning hormone which will have no function, or even have a bad function which will affect our body. So in this slide, we understood how the initially the protein, once it's translated, it is modified and packaged and sorted inside the Golgi and the smooth endoplasmic reticulum region, and then the immature secretory granules can be transformed into mature secretory granules and can be released. You need to understand that some of the granules can be secreted straight away in the case of immaturing, because it needs some other further steps to be accomplished either in the bloodstream or in the target organ. So continuing to talk about the classification of hormone according to chemical composition. So the last 2 slides or 3 slides were describing the classification of hormone according to as well chemical composition, but they are the polypeptide hormone. Here there are some other types of hormone which is called amine hormone or amine hormones, and these type of hormones are all derived from the amino acid tyrosine. We have the thyroid hormone, we have the adrenaline, we have the noradrenaline, and we have the PIH. The third type, according to chemical composition, is the steroid hormone. And these hormones are all derived from cholesterol. And of course, these steroid hormone include the sex hormone from the gonads and placenta, all the adrenal cortex hormone, which come from the adrenal part, or specifically the cortical region of the adrenal gland. And we have as well the 125 dihydroxyvitamin D3. So again, according to chemical composition, if you want to summarize, we have the chemical type, either a peptide or a protein, and this of course is formed from many amino acids. One of the major examples is the oxytocin hormone, and you can see that it is composed of around 8 amino acids, and this is the structure of oxytocin. And since it is composed of amino acid, that's why it's called peptide or oligopeptide hormones. The second as well, as we said, is the amino acid derivative hormone. And you can see that it is synthesized, generally it's synthesized from 1 or 2 amino acid max. The major example is the norepinephrine and the thyroxine hormone. The third type, as we said in the previous slides, is the steroid hormone. That is generally, it is synthesized from the cholesterol. And you can see the famous example is the sex hormone, including the testosterone and the estrogen. And of course, you don't need to memorize the chemical structures of these hormones. The third criteria of classification of hormone is based on solubility. So you have a water-soluble hormone and you have a fat or lipid-soluble hormones. Of course, the water-soluble hormones are all peptide hormones and all amine hormones, except the thyroid hormones, while the lipid-soluble or the fat-soluble include all the steroid hormones. So what about the mechanism of hormonal action? Actually, hormone, as we said before, hormone have to interact with the receptor on the target cell in order to affect that target cell. And these hormone receptor have two locations, either exist on the plasma membrane of the target cells or exist inside the cell itself, even in the cytoplasm or even in the nucleus. So, as we said before, there are hydrophilic hormone that can act two types of hormone, either water soluble or lipid soluble. So generally we have a hydrophilic hormone and hydrophobic hormones. So the hydrophilic hormone actually act and have an action and should bind on receptor that is on the plasma membrane, while the hydrophobic hormone acts only on the receptor that is called nuclear receptors. So we need to differentiate now between two different nomenclature of hormones. It's called the tropic hormone and non-tropic hormone. So what is tropic hormone? Actually, tropic hormone stimulates an endocrine gland to grow and secrete its hormone. So in human, the tropic hormone are secreted by the anterior pituitary gland. And there are 4 types of these tropic hormone, including the TSH, FSH, LH, ACTH, adrenocorticotropic hormones. So all of these are secreted by the anterior pituitary gland, and they are the tropic hormone. While the non-tropic hormone, they target and stimulate non-endocrine tissue to induce effect. So they actually produced as well by the anterior pituitary gland, but different hormone, including the prolactin and MSH. Some hormone, like the growth hormone, have both effect, either tropic or non-tropic. So to summarize the difference between the tropic and non-tropic, majorly the non-tropic, it will affect a non-endocrine tissue, while the tropic hormone, it will affect an endocrine gland to grow and secrete its hormone. So basically, the hormone is secreted from an endocrine gland in the tropic hormones. The hormones are secreted from an endocrine gland and will affect another endocrine gland. While the non-tropic hormone, it target and affect a non-endocrine tissue. The famous example that combine both actions, tropic and non-tropic, is the growth hormone. It can have effect on endocrine tissue and non-endocrine tissue. So continuing the mechanism of action, so as we said before, based on the type or the solubility of the hormone, the type of receptor it needs to interact. So we have different types of human receptors, hormone receptors. Receptors could be on the surface of the cell membrane. Receptors could be in the cytoplasm. Receptors could be in the cell nucleus. And of course, we'll deal each of these receptors in details. The famous example for receptors of the cell membranes are including hormone which are protein in nature and catecholamines hormone, while receptors that deal with in the cytoplasm deals with the steroid hormone, while receptors in the nucleus deal with the thyroid hormones. So you need to understand that the number and sensitivity of these hormone receptors are regulated. You don't need to, cells don't need to express a lot of these receptors, and as well don't need to express few of that receptor, because expressing a lot of receptor will need a lot of molecules of the hormone. It will trigger an upregulated action of the hormone. So the number of the receptor, either on the surface, in the cytoplasm, or in the cell nucleus, should be regulated in the target cell, and usually do not remain constant. And usually, if they exceeded the number of molecules of this receptor, if they exceeded a certain number, they have to be down-regulated. Okay, they have to be down-regulated in order not to have an overaction of the hormone once it bound to the receptor. So this down-regulation of this receptor can occur as a result. Why the target cell, which have the receptor, need to down-regulate the number or decrease the number of that receptor. So the down-regulation occur actually either by inactivation of some of this huge number of the receptor molecule, inactivation of some of the intracellular protein signal molecules. In order to activate the receptor either on the surface or inside the cell, it needs some molecules to activate that. So in order to down-regulate this receptor, you can inactivate some of these proteins that convey the signal to the receptor. Some cells do sequestration of this receptor. And what's the meaning of sequestration of this receptor? You can take the example of a receptor if it's exposed on the surface of the cell. If it's exposed on the surface of the cell, once there is a hormone, it will bind to that receptor. So one of the methods of the down-regulation of that receptors is to pull down this receptor from the surface and enclose it in a vesicle inside the cell. So this process of temporary sequestration, it prevented the contact between the receptor and the signal, which are the hormone, in order to down-regulate the number of the receptor on the cell surface. As well, another method of down-regulation of the hormone receptors is to destroy the receptors by lysosomes after they have been internalized. And of course, the most famous thing to down-regulate the receptor is already to decrease the production of these receptors, because this receptor will be as well protein in nature, so it needs to be transcribed and translated and got exposed either in the cell surface or in the cytoplasm. In each case of these 5 cases, the receptor down-regulation decreases the target tissue responsiveness to the hormone. And of course, this is very important in order to regulate the action of the hormone toward its target cell. شباب، حد عنده سؤال في الجزء ده في السلايد ديت؟
نو دكتور، مش الحال.
طب حد يقدر يقول لي احنا ليه بنعمل داون ريجوليشن للهرمون ريسبتورز؟ او ليه الخليه ساعات تبقى محتاجه تعمل داون ريجوليشن للهرمون ريسبتورز؟
لحتى ما يصير في عنا اوفر برودكشن.
اوفر اكشن.
اوفر اكشن اوف ذا هرمون.
مش يعني اوفر اكشن، لان الهرمون كده لما هيطلع في الدم لو لقى الريسبتور هيعمل الاكشن حتى لو جسمنا مش محتاج الاكشن. فهمتوا؟ اوكي. فبالتالي الخليه بتدور على ميكانيزم ان هي تمنع الريسبتور ان هو يبقى موجود على السطح او موجود في السيتوبلازم عشان يتعرف عليه هرمون. فهمتوا؟
يس دكتور.
اوكي. طيب يا شباب، اوكي، that's good. So now after we have said that there are 5 mechanisms to regulate the number and sensitivity of hormone receptors, we need to understand how the mechanism of hormone action. So we'll start with the mechanism of the hormone action of steroid hormone. So generally steroid hormone are hydrophobic and as well lipophilic. They act on nuclear receptors, not on the cell surface receptors. So they function majorly, this steroid hormone, to change the protein synthesis of other transcribed genes. So actually the mechanism of the steroid hormone is called direct gene activation or direct messenger mechanism, in which the hormone diffuse, because it's lipophilic, it will diffuse straight through the plasma membrane. It will bind and activate the nuclear receptor. This nuclear receptor, although it is called nuclear receptor, but it exists in the cytoplasm. Once in the cytoplasm will be bound with this steroid hormone, this hormone receptor complex will migrate to the nucleus and start to activate the gene for transcription into messenger RNA. Soon after the messenger RNA is formed, this messenger RNA will migrate into the cytoplasm to be translated into protein. So the signal starts with a hormone that have succeeded to penetrate the cell membrane and bound to the receptor, to the nuclear receptor. We need to understand that this intracellular receptor or the nuclear receptor, they function as transcription activators, and they exist in inactive state in the cytoplasm, this nuclear receptor. And once they are bound to their ligand, which is the hormone coming from outside of the cell, it will be active. And as we said, it will migrate into the nucleus and behave as a transcription activator for other genes. So let's see here a diagram representing the what we have just said. So initially you have a steroid hormone, as you can see, outside the cell. Initially you have a steroid outside the cell, and it succeeds to penetrate inside the cell cytoplasm. And in the cell cytoplasm, you have a nuclear receptor. At this stage, it is inactive. Once the receptor hormone complex is formed, this receptor hormone complex, or the ligand nuclear receptor complex, it will migrate through the nuclear bore inside the nucleus, and it will bind to a specific region on the DNA, and it will promote the transcription of a specific gene into messenger RNA that soon it's transported outside to the cytoplasm to generate a new protein. So these are basically a summary of the mechanism of action of steroid hormone. So again, the steroid hormone diffuses through the blood membrane, binds to the intracellular receptor, which is called nuclear receptor. This receptor hormone complex will enter the nucleus, that will bind to the DNA region, will initiate transcription of a specific gene or a certain gene, and this messenger RNA directs protein synthesis of the new protein. So that's why it's called a direct gene activation, because the steroid hormone leads to the activation into the transcription of a specific set of genes. And that's why as well that the receptor hormone complex is generally is called the transcription activators, because it activates transcription. Let's talk up here about some of the nuclear receptors. So we are talking here about some examples of these nuclear receptors. So you can see that a lot of the nuclear receptor, it shares conservation in their amino acid sequence, and they have majorly 3 functional regions that are the same in different receptors. So here you can see 5 examples of the nuclear receptors, which are the estrogen receptor, progesterone receptor, glucocorticoid receptors, thyroxine receptor, retinoic acid receptor. And of course, these receptors are named according to the ligand or the hormone which will bind to that nuclear receptors. Generally, you can see that all these 5 receptors, although they are different in the number of amino acids they form, these are proteins, you can see that the estrogen receptor consists of 553 amino acids, the PR consists of 900 something, 700 something, 408 for the thyroxine receptor. Although they are different in the number of amino acids, but sequence and the number of amino acids they formed, but all of them share majorly three functional regions. And this is the general primary structure to understand that most of the nuclear receptor have a conserved functional region in their structure. So you have a variable region, you have a DNA binding region, and you have a ligand binding region. So basically the DNA binding domain, this one, the blue one, this one helps this receptor to bound to DNA, while the ligand binding domain help, that means the amino acid sequence of that region help the receptor to bind to its ligand, to bind to its hormone. And this variable region have a different function, and it is variable among the receptors. So the variable region, part of it may function as activation domain. So it activates the complex. While as we said, the DNA binding region or domain has a repeat of C4 zinc finger motif, and this enables this complex to bound to DNA. While the ligand binding as a domain, as we said, it functions as a repression domain in the absence of the ligand. So once there is no ligand, as we said before, the nuclear receptor have to be inactive in times that there is no hormone inside the cells, in order to not overactivate the gene activation, in order not to produce protein that is not necessary for the cell. So this ligand binding domain, it inhibits the activity of that receptor in order not to migrate toward the nucleus. So once there is a ligand that is internalized inside the cell, it will bind to this ligand domain, and it will activate this nuclear receptor to be translocated inside the nucleus. فاهمين يا شباب الفانكشن بتاعت الثلاثه ريجنز دول ولا لا؟ شباب صباي؟
Yes, doctor, it is clear.
اوكي كلير اوكي.
دكتور
ايوه يا فندم اتفضلي.
بس اللايجند بايندينج دومين ما فهمت كتير أنا كيف هي it acts as a repression domain إذا ما عنا لايجند.
اوكي انت فاهمه ان الهرمون لما بيخش السيل بيعمل ايه؟ الهرمون ده او الليجند ده بيعمل ايه؟
هيعمل بايندينج.
ممتاز ممتاز. افرضي ان النيوكلير ريسبتور ده كان مش كان اكتف without the هرمون، ايه اللي كان هيحصل؟
ما فهمت دكتور السؤال كيف؟
What if this nuclear receptor is active without the need of a ligand, without the need of hormone? What will occur?
كتير عنا over action لهذا
It will migrate to the nucleus and will activate genes to be transcribed. صح كده؟
yes صح.
طيب فاصبحت النيوكلير ريسبتور دي عندها جزء منها اللي هو الليجند بايندينج دومين. بيعمل ايه الليجند بايندينج دومين؟ يعني هوريكم I can explain. شوفوا الرسمه. Let's assume ان الاستراكشر اهو اللي انتم شايفينه ده قدامكم 3 ريجن. هو الطبيعي في الخليه بيبقى عامل بالمنظر ده. لو افترضنا ان هو كده اهو. ادي ريجن، ادي الريجن اللي هي الخضراء، ودي الريجن اللي هي الزرقاء. الريجن الثانيه بتبقى عامله كده. دول الثلاثه ريجن. الريجن الثالثه دي هي دي الليجند بايندينج دومين. فهذه الليجند بايندينج دومين بتعمل ايه؟ بتعمل inactivation للريسبتور. once there is a ligand ده هيتفتح كده. ادي الثلاثه ريجن. والمنطقه الاخرانيه اللي هي الليجند بايندينج دومين دي will bind to the hormone. مثلا for example once it bound to hormone, the whole complex which is the receptor with the ligand will be active, so it will go inside for the nucleus to do the activation. فبالتالي اصبح ايه؟ اصبح هذا الليجند بايندينج دومين دوت اصبح in the absence of the ligand it will act as an inhibition for the nuclear receptor in order not to go in order not to go inside the nucleus. فهمتي؟
ايه اوكي. ايه انه لانه ما في الهرمون اكيد ما حيصير في بايندينج من الاساس، يعني ما في شيء يصير.
تمام. بالاضافه لذلك ان هذا الليجند هو بيعمل انهبيشن للنيوكليير ريسبتور ده في عدم وجود الليجند. فهمتي؟
اوكي
طيب نكمل. Let's see one of the very famous examples that describe the importance of these regions of the nuclear receptor. As we understood, there are three regions. The three regions are, again, the variable region, the DNA binding domain, the ligand binding domain. So we need to understand which of these regions are important for the process of translocation of these hormone receptor complex into the nucleus. Which of these ligand is responsible for this translocation? So let's see here, for example, we will use in that experiment a protein, although it is not related to hormone at all. It's called beta-galactosidase. What is the importance of this hormone, of this protein? It's not a hormone. What is important of this enzyme or this protein? Actually, this enzyme inside the cells can emit fluorescence. So you can see the localization of this protein inside the cell. So once we put a signal, which is called DAX here, represented by DAX, this signal is a steroid signal. So we assume that this signal will penetrate the cell membrane and got internalized. And once it penetrates inside the cell membrane, so it needs a receptor. So we used this protein, as we said, because it emits fluorescence. We can see its localization. So you can see here its localization represented by the bright light. So you can see it is localized in the cytoplasm because the bright light is outside in the cytoplasm of the cell. While the nucleus appears dark, that means this protein is only localized in the cytoplasm, whatever there is a steroid signal or there is no steroid signal, minus there is no steroid signal, plus there is steroid signal. So this is like a control experiment that say that the beta-galactosidase is localized in the cytoplasm, whatever we use a steroid signal or we don't use a steroid signal, because actually beta-galactosidase is not a nuclear receptor. So this DEX will not bound to this beta-galactosidase. Again, why we use beta-galactosidase? Because we can see the localization of that protein through the fluorescence that it emits. So how we can use this beta-galactosidase protein to see the movement of the nuclear receptor from the cytoplasm to the nucleus? Let's see. So here you can see here, we will fuse or bound by genetic engineering. We can bound this beta-galactosidase region with one of our nuclear receptor, which is the glucocorticoid receptor. And you can see that these glucocorticoid receptor have the free region we just mentioned in the previous slide. And now, although we can't see the glucocorticoid receptor by itself, but once this glucocorticoid receptor is bound, once this glucocorticoid receptor is bound to the beta-galactosidase, it will emit fluorescence because they are fused together. So again, if we treat cells with the steroid signal represented by DEX, you can see that the nuclear receptor, which is fused to the beta-galactosidase, will localize inside the nucleus. How did we know that it is localized inside the nucleus? Because it is fused to the beta-galactosidase, which emit fluorescence. So it will be shown to be inside the nucleus. In cases that there is no steroid signal or no steroid hormone, you can see that the glucocorticoid receptor are existing in the cytoplasm. So here we can see, Mayer Rifai, could you mute your mic, please? So you can see here the effect of the steroid signal or the hormone, the DX, in affecting the localization of the glucocorticoid receptor. But we need to know which region of these three regions actually controls this translocation from the cytoplasm to the nucleus. In the third setup, you can see now we fused the beta-galactosidase, which emit fluorescence, with only the ligand binding domain, not the whole three regions of the glucocorticoid receptor. We only fused with the ligand binding domain. And you can see here, when this is being induced by the presence of the DX with the DX steroid hormone, the complex is inside the nucleus, while in the absence of the DX, it is still in the cytoplasm. So from this setup, we conclude that the region of the nuclear receptor, or generally the nuclear receptors, the region responsible for the process of translocation from the cytoplasm to the nucleus, actually it is the ligand binding domain. So once the ligand binding domain bound to the ligand or the steroid hormone, it will move inside the nucleus. And you can see here the nucleus is bright in color in the presence of DEX. While in the absence of DEX, in the absence of the hormone, this ligand binding domain exists in the cytoplasm. Any question about this setup or this experiment? Okay. So now we have talked about, as we said, the mechanism of action for steroid hormone. Now we'll talk about for non-steroid hormone. So for non-steroid hormone, these hormones are hydrophilic or ribophobic, and actually they don't act on the nuclear receptors. They act on receptor on the plasma membrane. The famous example are amines, any amino acids, peptide, protein, glycoproteins, hormone, and they actually function in a mechanism, not a direct mechanism as the steroid hormone. It functions as a second messenger mechanism. In this second messenger mechanism, basically the hormone binds on the plasma membrane receptor. The receptor is activated. It will activate an enzyme, which is called the effector enzyme. Once this enzyme is activated, a second messenger is generated inside the target cell. So through the process of a cascade of reaction after this activation by the second messenger, a metabolic response triggered inside the target cell. So that's why it's not direct, because you need a second messenger, as we will see later in the few slides, So to summarize the effect of a non-steroid hormone, if you assume this is a plasma membrane and this is the extracellular and this is intracellular, you have a receptor, for example, like the insulin receptor. In this state, this receptor is inactive. Once there is an insulin, this insulin, it will bound to the receptor, leading to dimerization of the receptor. You can see it is dimerized. And in this case, this receptor will be active. Once it is active, as we said, this receptor will be activated, like step number 2. The activation of this receptor is by its own phosphorylation. So there is a phosphorylation signal in the cytoplasmic part of that receptor. Once this receptor is activated, it needs to activate another molecule, which is called effector enzyme. So you can see here that in another protein should bound to the activated receptor. Once it's bound, this receptor, this activated molecule, it will provide a cascade of reaction until reaching the response of the target cells. To give a very clear example about the process of activation or hormone action for non-steroid hormones, we will take 2 examples. So the first example, you can see here, receptor bound to You can see receptor here bound to the signal. And of course, as we said, this signal is lipophobic, so it cannot penetrate the cell membrane. It needs a plasma membrane receptor. Once this receptor, once this ligand, this hormone or growth factor bound to the receptor, this receptor will be activated, leading to the activation of one of the famous protein, which is called G protein. This G protein will soon got activated and activate another protein inside the plasma membrane, which is called phospholipase C. So once this effector enzyme is activated, it's a phospholipase. So it needs to break down something because it's a lipase. So basically, there is another protein, which is called TAPI protein. This TAPI protein is bound to one of the molecule of the plasma membrane, which is called PIP2, phosphoinositol diphosphate. So normally, the TAPI is inactive once it bound to PIP2. So the effector phospholipase C, once it is active, it will break down PIP2 into IP3 and DAG. Once PIP2 is broken down, this will lead to the release of this TAPI protein. So now this TAPI protein is released in the cytoplasm. One of the important characteristics of this TAPI protein, when it's got released of the cytoplasm, it has two domains, majorly two domains. A DNA binding domain, which will enable its binding to the DNA, and it has a transactivation domain, which will facilitate the activation of transcription of a specific set of genes. So once it is released in the cytoplasm, it will be translocated into the nucleus and bind to the DNA by the DNA binding domain and activate transcription by the transcription activation domain. And of course, it will affect the transcription of another set of genes. So in this example, we describe how the effect of hormone that can bound to the surface plasma membrane, leading to the activation of effector molecule, which is a phospholipase C. This phospholipase C will break down PIP2. This breakage of the PIP2 will release the TAPI protein into the cytoplasm. Once the TAPI protein is released in the cytoplasm, it will be translocated inside the nucleus, where it bound to the DNA and initiate transcription of specific genes. So what is TAPI protein? Actually, scientists have found that TAPI gene actually expressed primarily in brain, and the gene of this protein actually is involved in the control of eating behavior and involved as well in obesity. So how did we know that? Because they mutated this TAPI gene in mice. They found when they mutated this gene in mice, this gene developed obesity. So there is a malfunction in the process of if this gene is mutated, individual will get obese. So currently they are working on that gene to control obesity. And of course, this is not the only cause of obesity. Any questions يا جماعة حوالين الطبي gene؟ Okay. So this is one of this is the first example to show how the signal is transformed from the plasma membrane until reaching an effect inside the nucleus. So we need to understand here, where is the second messenger? Actually, the second messenger is PIP2, because once it is broken or degraded by the phospholipase C, it will release the TAPI protein to translocate inside the nucleus. Let's talk about another example, which is called GPCR, how GPCR is activated and trigger as well another pathway that control the activity of one of the famous enzymes inside our cells, which is called BKC, and as well controls the amount of calcium inside our cells. You need to understand that calcium ions, one of the very important ions that control a lot of function inside our cells. So again, the receptor is activated. That's why this pathway will be called IP3 DAG pathway. So you can see here the receptor as well is got activated. It will activate a protein which is called G protein. That's why this receptor is called G protein coupled receptor, GPCR. Once activated, once this one is activated, it will lead to activation of the same enzyme, which is phospholipase C. And of course, phospholipase C, as we said in the last slide, will work on PIP2. Once it breaks down the PIP2 into diacylglycerol, DAG, and IP3, you have two different routes for these two molecules. The IP3 will move in the cytoplasm. It will bound to a specific channel, which is called IP3 gated calcium channel. Why it's called IP3 gated calcium channel? Because this calcium channel control the flow of calcium from the endoplasmic reticulum to the cytoplasm. And in order to be operated and in order to be open for the calcium to go outside from the endoplasmic reticulum to the cytoplasm, it needs the IP3 molecule. So once the IP3 molecule is bound to, once this IP3 molecule is bound to the channel, it will open the channel for the calcium to go out. Once the calcium is out in the cytoplasm, it will be bound to the BKC. It's one of the famous kinases inside the cell, which is called protein kinase C. This protein kinase C, once it's bound with the diacylglycerol DAG, which is initially formed due to the breakage of PIP2, it will lead to activation of this protein kinase C, which will lead to the phosphorylation of other substrate, which is important for a lot of other function inside the cell. So you can see here that there is a lot of signaling is occurring due to the effect of the signal that binds on the surface of the plasma membrane. So where is the second messenger here? You can see that the IP3 considered to be a second messenger. Okay. So let's see the full picture and what is the difference between the first messenger, the second messenger, the third messenger, and the fourth level. So initially you have a hormone, which is either hormone 1, hormone 2, hormone 3. This hormone will bind to the receptor, and each hormone will bind to its specific receptor. Each hormone will bind to its specific receptor. And each hormone, once it's bound with its specific receptor, it will activate certain molecule. In this case, it will activate calcium, a cyclic AMP, diacylglycerol, and this will consider to be the second messenger. Once this is activated, the diacylglycerol, as you can see, the diacylglycerol will activate BKC, as you can see here. This one is diacylglycerol. Together with calcium, it activated BKC to do the further function. So here as well, another example, cyclic AMP, it will activate another enzyme, which is called pKa. While the calcium, it can activate another kinase, which is called calmodium kinase, CMK. Anyway, after the activation of this third effector, which is usually is a kinase, this will be activate a set of transcription factors. One of the famous transcription factors, which is called CREB, and this transcription factor have the ability to bind to the DNA to induce transcription of another set of genes. So in this slide, we need to understand there is a lot of signaling pathway that involve the recognition of a signal from the surface of the membrane. And this signal is conveyed through a cascade of reaction until reaching to the DNA in order to activate the transcription of another set of genes. And of course, down here, you can see the nomenclature of each initials up there. The DAG, diacylglycerin, PKC, PKA, okay? In this last slide, we'll give a brief introduction about the hormones and how the homeostasis regulation. So hormone actually, as we understood now, transmit information about stages of one organ to another. So they regulate corrective action to maintain homeostasis. For example, when we have glucose elevated in our body, the pancreas will secrete insulin in order to lower this glucose amount in the blood. And this is a process of maintaining homeostasis. Insulin, of course, travels through circulation. It will signal liver and the fat cells to increase the permeability to glucose. So glucose will move from the blood to be stored in the form of glycogen inside our liver and our fat cells and even muscles. That will lead to a decrease in the blood sugar level in order to reach a homeostasis state. So to be effective for the hormone, to be effective, the hormone needed should not be degraded too quickly or too slowly. So why? Because if it's degraded too quickly, the hormone, if it's degraded in the blood too quickly, it will not perform the correct action. And if it's degraded too slowly, it will perform overaction. For example, in the example of insulin, if the insulin is not in the optimum concentration, if there is a lot of insulin, this will lead to a high decrease in the amount of glucose inside the blood, which may lead to a coma since the glucose is not reaching the brain. So that means if the insulin is not degraded too quickly, if it's degraded too slowly, that means there will be a lot of amount of insulin in our blood. This will lead to a lot of storage of glucose inside our tissues. This will lead to a high decrease in glucose in our blood, which may lead to a coma. As well, if it is not degraded too quickly, if it's too slowly, if it's degraded too slowly, as we said, it will increase the amount of glucose taken by cells. But the other way around, it will lead to inefficient storage of the glucose. So basically, it will have no function. As we said, if it's too quickly, it will not arrive at target cells because it will be degraded. If it's too slow, the information conveyed will be obsolete and may evoke an inappropriate response. So this example, as we said, for the insulin, but different hormones have varying half-lives. Some of them can exist for 3 minutes. Some of the hormones can exist for hours. Some of the hormones can exist for days. All of these half-lives in circulation, it depends in part on the distance that the signal have to travel in order to reach its target cell. So controlling the hormone secretion and regulation of the homeostasis, one of the important parameters that regulate the function of the hormone. Shabab, this concludes our lecture for today. Any questions?
No, doctor, thank you so much.
They cross the membrane.
معلش السؤال تاني
في
السؤال تاني اتفضل.
Yes, in steroid hormones عندنا when they cross the plasma membrane في nuclear receptor.
تمام.
ليه ما سميناه cytoplasmic receptor؟
ليه ما سميناه cytoplasmic receptor؟
Yes.
ايوه لان الاكشن بتاعه في النيوكليوس.
Okay, so its purpose انه بس to transport it من plasma membrane to the nucleus.
تمام. طول ما هو موجود في السيتوبلازم هو ملوش لازمه في عدم وجود الهرمون. فاهماني؟
Right.
اوكي.
Thank you.
You're welcome. في حد عنده سؤال تاني يا شباب؟
Yes, Doctor. الباثواي تبع الهرمونز we are required to memorize.
Yes.
Okay, thank you.
في حد عنده سؤال تاني يا شباب؟ في حد شايف ممكن نغير الشرح بطريقة تانية ولا it's good? It's almost our second lecture today. صح كده؟
Yes.
في حد محتاج نغير من طريقة الشرح، محتاج نعمل حاجة تانية؟ برضو أنا بحب أسمع الفيدباك بتاعكم.
No, so far so good.
أوكي. لو حد عنده تعليق يتفضل يقول. أوكي يا شباب. Thank you and see you next week.
Thank you, doctor.
Thank you. Bye.